Thank you for the introduction and thank you for coming here. And thank you for the invitation to come and tell you a bit about my work. I've had a great day talking to lots of good students, very motivated, enthusiastic students about population genetics. So it's been really, really fun. Yeah, so like Robert said, I'm going to be talking about Trinidadian guppies and specifically what they can teach us about rapid and repeatable evolution. So this is a little picture of guppies at the bottom there. And the tape, so those who were born maybe in the last 30 years might not know this. This is a cassette tape, and it's referring to Stephen Jay Gould's thought experiment that if we replayed the tape of life, would we expect to see the same thing? And his argument was no, there's too many chance events, contingencies, too many just random things happening in the tape of evolution. But in response to this, Simon Conway Morris saw his arguments and he said, well, we don't need to imagine replaying the tape of life because we can look to convergent evolution, so repeatable evolution, where individuals solve the same sort of evolutionary problems with similar solutions. So just so we're all on the same level, I'm going to use the words convergent evolution to mean any sort of repeatable evolution, and parallel evolution when I actually mean sort of same parallel trajectories. So we can think of convergent evolution as these big macro evolutionary events, something like wing shape in dinosaurs, bats, and birds. They've all evolved wings to fly, or complex lens-based eyes in annelids, cephalopods, and vertebrates. They've all evolved these same shaped eyes. But we can also think of sort of microevolution type events. So you can think of sort of within species rapid evolution. So a classic example of this would be the marine and freshwater stickleback. So freshwater stickleback having these loss of armor plates, repeatedly adapting to freshwater environments over and over again, or these pocket mice that have evolved fur color to match their backgrounds over and over again in different environments. So why are we so interested in this repeatable evolution? And if selection is repeatable, then we can start to predict it. And if it's not, then we might be able to predict when it's not repeatable. So for me, the real big questions are, why is it repeatable sometimes and why not? And we can think of things like constraints. So is evolution forcing the path of evolution down specific trajectories? We can think of contingencies, so these chance events that happen in the course of evolution. We can also think of time scales. So is early evolution different than from long-term evolution? So how populations adapt quickly to the environment. And to answer those questions, I argue that you really need to link selection to genotype and phenotype in the wild within this within one species. And you kind of need a species that varies with all these types of parameters to really answer this question: is selection repeatable, and is it therefore predictable? So lucky for me, we've got a great system where we've already got that link from selection to phenotype, and we've got variation within the system. And that system is the guppy system. So I've been working on the guppy system now since my PhD. That picture at the top there is me collecting guppies for the first time during my PhD. And they exist in these headwater streams in northern Trinidad. Beautiful, beautiful area. And these headwater streams are separated by waterfalls. And the waterfalls are really important in the guppy system because below the waterfalls, guppies coexist with predators. So like this pike cichlid here, seen at the bottom. The predators can't get above the waterfalls, but the guppies can. So we've got high predation below the waterfalls and low predation above the waterfalls. And there's been work going on since the 70s and 80s and even earlier that have compared this high predation, low predation dichotomy within a river. And the first work was really done on male coloration. So on the top here, we've got a low predation guppy. He's got these big bright orange spots. And on the bottom, we have a high predation guppy, so much more drab. And you can see how that would relate to predation and sort of evading predation, but then also sexual selection in the low predation environments. So like I said, I do this work in northern Trinidad. So if you don't know where Trinidad is, it's just off the coast of Venezuela, here in the little star. And we do it in the northern part of the range because the northern part of the island is because the northern part of the island has this mountain range. So what you have obviously is rivers going over waterfalls because they're going over mountains. But what you also have is this separation of rivers from each other. So one low predation population in one river is separated by a whole mountain to the next low predation population. So you can't get that migration from low predation to low predation. It has to sort of go all the way down and back up again. So what we have then is this independent adaptation replicated across river and river. And so as scientists, we love replication. And this one is really a natural replicated system across the northern part of Trinidad. So since the work of the 70s and 80s by John Endler and collaborators that looked at color, there's been a whole host of phenotypic traits that have been studied in this low predation and high predation dichotomy. So we've got color, so we know low predation guppies have more diverse, bigger, brighter spots than high predation guppies. There's also a whole suite of life history traits that are different. So low predation guppies are bigger, they mature later, at a bigger size, they give birth to fewer and bigger young than high predation guppies. And then there's also differences in behavior. So differences that you would expect to be related to predation. So low predation guppies court more, and high predation guppies shoal more, faster escaping, all that sort of stuff that you would predict based on this high predation and low predation dichotomy. The other really exciting thing about the guppy system is that we have in situ experimental populations. So this is where researchers have taken guppies from a high predation locality and found areas of the rivers that even they can't get to. So even guppies can't pass these waterfalls. So they've translocated them to these new environments, these new low predation environments, and they can document phenotypic evolution over time. And so what they found is that phenotypic evolution was documented fairly quickly, within 4 or 5 years. So guppy generation times, about 2 to 3 generations per year. And that the introduced populations resemble naturally colonized low predation populations. So here is just work done by David Resnick on male size with 2 such introductions. So we have Arepo and El Cedro introductions, and they're bigger than their source population. So we've seen this response fairly quickly over time. So there's been a series of these introduced translocation experiments. The first one in 1956, the next 2 in 78 and 81, which I talked about here, and then a much more recent one in 2008 and 2009, and they've occurred across the island. Right, so we've got quite a bit of information of how selection acts on the phenotypic side, but really, we're not really understanding how selection is acting at the genotype. So what my work does is kind of fills in that other half of the triangle. So how is selection acting at the genotype, and how does that influence evolution? So big questions we're interested in are, does phenotypic convergence equal genomic convergence? So do we see repeatability at the genetic level? What's the importance of starting genetic variation? So these are kind of like the contingency events. You know, what do we have to work with to start? We can look at the importance of time scale because we've got these introduced populations. And then more recently, we can start to look at polygenic versus large effect loci response. So those are sort of the big questions that I'll talk about today. I'll go through sort of three main studies that we did, all using whole genome sequencing and population genomics in guppies. So the first one I'll talk about today is comparing natural high predation, low predation pairs, and that was done by Jim Whiting. Then I'm going to talk about that very early, the most recent transplant experiment. So those were the ones introduced in 2008, 2009. And that was work done by Micah Vandersee. And then I'm going to talk a little bit about some temporal genomics that we have in prep today. All right, so jump right into it. So the first big question, does phenotypic convergence equal genomic convergence? So again, we're looking at high predation, low predation population pairs, and we've sequenced from 6 different rivers across the island. And like I said, this is work mainly done by my past postdoc, Jim Whiting. So we've got these 6 rivers, and each one a high and low predation pair. Probably this is too much explanation for people who study population genomics, but I'm just going to go into it a little bit. What most people do when they're looking for replicated or convergent evolution is something called the overlapping outlier approach. So this is when you've got multiple replicates. So for us, we have high predation, low predation. So maybe the red is the high predation, the low predation is the blue. You could have a common ancestor. So maybe the marine stickleback to lots of freshwater, lots of comparisons, Kleins. Whatever your sampling design, your overlapping outlier approach is quite similar. You take some measure of differentiation, probably FST or something like that. You scan the genome and you're looking for peaks. So where along the genome are your populations differentiating and where do they overlap? So that's the stronger evidence of selection is where they're repeatedly showing signatures of selection. So that's generally the overlapping outlier approach, and we're going to take that today as well. Right, so just to start doing some basic population structure, we've got our 6 river pairs, and we see what we expect genome-wide is that the river pairs cluster together on the phylogeny or using a structure plot like this. So each one of these tiny squares is a low predation to high predation, and then they cluster together in a bigger pair, or you see that as well at sort of the general phylogenetic approach. What becomes important later is that there's also clustering within river drainages. So we've got this major drainage called the Caroni in the west. So these 3 rivers all flow into the bigger Caroni River, and they all cluster together. And then in the east, we've seen this cluster of the eastern drainage, the Orapooch, and northern. So we've kind of got this broader clustering as well, which becomes important when we're looking at starting genetic variation. So to start, we're just going to look at our 6 pairs and try to find outliers. So the overlapping outlier approach reveals really little repeatability. So this is an upset plot at the bottom here. So it's just showing how many outliers in each of the 6 comparisons, and then how many shared by 2, how many shared by 3, and then on and on. We don't see anything shared more than by 3 rivers, and even then it's quite a minimum amount of windows. So what we would say that at this point is that there's been very little replicated evolution genome-wide. But we can take these outliers and see what pathways they're in using KEGG pathways, and to see if any pathways are enriched repeatedly in our multiple drainages. And we find some pathways enriched, but importantly, we find one pathway that's enriched in all six population or six river pairs, and that's the Cadherin pathway. So we've got some evidence of more pathway level repeatability at the genome in these pairs. We've also taken what's called a Bayes approach. So this is correlating allele frequency environment. So this is putting all your data together in an allele frequency matrix and an environmental matrix and seeing if there's correlation. And so this is the data across the genome, and we see two really big peaks, one much bigger than the other, on chromosome 8 and chromosome 20. So here there's a real strong candidates for allele frequency change correlated with environmental differences. So a better evidence for repeated selection. So on chromosome 8 and chromosome 20. So chromosome 20, let's go into that in a bit more detail. So that's chromosome 20 at 2 MB. This on the left is a genotype plot. So in blue, we have the alternative haplotype. Red is heterozygote and yellow is the reference haplotype. And so you do see this big block of individuals that are fixed for this alternative haplotype. And they tend to be from the low predation environment, but just in one drainage. So if you remember, we've got the Caroni rivers flowing to the west. In all those three low predation populations, we find this fixed or nearly fixed alternative haplotype. And that you can see a bit more in this phylogeny here, where we've got mostly the triangles, the low predations clustering together, and then just pretty much everyone else in the clustering for the rest. So we've got a better evidence of repeated selection, but it's much more dependent on drainage structuring. So starting genetic variation, standing genetic variation is probably driving this differences in haplotype structure or haplotype response. So to conclude that paper, we can say sort of genomic convergent evolution is limited across all of our pairs at individual sites. But we see some convergence at the pathway level, so that Cadherin level. We've got one good candidate for adaptive loci at chromosome 20, but this really depends on your starting genetic variation. So if you started with similar genetic variation, then you're more likely to show a similar response. What we were left at the end of this study was really this question: can we be better at detecting convergent evolution? Because it seems like we're not getting all the whole story using this overlapping outlier approach. So what Jim did is he came up with a multivariate approach to detect parallel evolution. So like I said, most studies look for selection first, look for their outliers, and then try to overlap them among replicates. But what if we first looked for parallel change and then saw what individual sites are doing? And what if we extend that a bit further and look for things like parallel and anti-parallel and multi-parallel change? So this is really where I think this method comes into its own to be able to find different trajectories of change. So Jim called this AF vapor. It's an Egan analysis on allele frequency vectors. So each window along the genome, we're trying to find strong Egan vectors that explain most of the variation. And we're trying to find where there's parallel change versus multi-parallel versus completely divergent. A bit complicated, so I'll go into a bit more of toy examples by what I mean. So parallel means these allele frequency vectors are changing in the same direction. So, and on the same eigenvector. So we've got the top are all our parallel, But we can also do anti-parallels. So they're on the same trajectory, but they're going in opposite directions. So we can detect that with this method. If you've got more than 4 replicates, you can also do things like multi-parallel. So is there a strong one direction of change for, say, some of your replicates and a strong common direction of change for another set of your replicates? So looking at sort of multi-parallel and getting more at that question of real convergence versus divergence at the allele frequency level. So if we apply that to our populations, sorry, that should say 6 HPLP pairs, we see some nice outliers on eigenvector 2. So we see nothing on eigenvector 1, which is kind of expected because we didn't see any parallelism earlier. But we see some nice outliers on eigenvector 2. So that's a test of multi-parallelism. So we've got some of our replicates going in different directions, but at the same loci. What's nice is chromosome 20 pops up again. So we're confident that's still under selection. But another region pops out as more significant, and that's chromosome 15. And we've found that that area has been previously identified as selection in a few river pairs using both whole genome sequencing and my old work on RAD-seq sequencing. So it's nice to see some confirmation of selection happening with this eigenvector approach. Okay, so moving on, I wanted to talk about some of our rapid adaptation work. So this is a new experimental transplant system that was transplanted in 2008, 2009. And what's nice about this transplant experiment is that it's replicated. So we've got 4 sites. So these are our 4 sites here in pinks and purples. And we've got a phenotypic response already documented. So these sites are monitored over time using mark recapture experiments, but also once in a while they bring them back in the lab and do common garden experiments to see if there's a phenotypic evolution. And we do see a response in size and a response in color in all 4 replicates. So what we were interested in, what Micah has done, is to look at the genomic response. So how does it compare to naturally colonized rivers? And what are the effects of known demographic change? So to explain that a bit more, is like I said, these populations are monitored through mark recapture data. So we've got census size. So this is our census size for each of our 4 populations over time. So you can see each of the 4 populations have grown. But there's also differences in sort of random fluctuations. Specifically, this last one here, the Taylor population, had crashed in 2010 and has since grown. Okay, so we've got known phenotypic response, known demographic changes, what's happening at the whole genome level. So first, introductions still mostly resemble their source. There hasn't been a massive change genome-wide at the genomic level. So this is just our river pairs again, and I've just added in our introduction experiments here. And they cluster with a source. If we zoom in PCA space, you can see that they're all sort of slightly diverging from each other, but overall still very similar to their source. We also looked at measures of diversity, so pi, Degina-Z. This one is expected heterozygosity of each of the transplant experiments compared to the source population is generally the same. So we're not seeing this massive effect of bottlenecks or founding events, which is nice, sort of genome-wide SNP level. But we do see some signals of bottlenecks using runs of homozygosity. So here we're just looking to see if there's long haplotypes of homozygosity across the genome. And if you have a lot of them and they're very long, this is an indication of inbreeding. So in all 4 of our populations, we have more runs of homozygosity and longer runs of homozygosity than the source. So the source is GHP here and to the side. And that Taylor population that had that massive bottleneck, that massive crash, has the longest and the most runs of homozygosity. So it's nice to be able to see the effects of demography replicated at genome-wide. So what about selection? So this, we looked at both allele frequency change and haplotype statistics. At just general allele frequency change, there was not a lot of fixed differences. There was no strong signal of repeatable fixed differences. But if we look at haplotype statistics, so this one is IHH12, we start to see outliers, and we start to see outliers shared by all our replicates. So specifically, just wanted to point out chromosome 15, 5MB, we've seen it before, and we're seeing it again at 3 of the 4 replicates there. So something's happening at that level, at that loci in the genome. We also looked at using our AF vapor, using our multivariate statistic, and we find lots of windows showing full parallelism allele frequency change, 65 windows across the genome. So possibly there is some evidence of polygenic change there. But the biggest change, or sorry, the sort of most striking change is again chromosome 15 at 5MB is a nice big chunky outlier. So full parallelism happening. So even though we didn't see it we only saw it in 3 of our 4 replicates, if we do full parallelism, we see it coming out. We've also compared parallelism to simulated neutral change, and that's what's shown on the right there. And chromosome 15, again, pops out at 5MB really strongly, different from neutrality. So just to remind you that that area of the genome has been identified before in a few rivers, a few naturally colonized rivers, as well as being multiply parallel in other rivers. And it overlaps a cadherin gene, which is a nice similarity between our natural populations. It overlaps these beta cadherin genes, which may have something to do with pigment cell migration, but we don't know. So to conclude that work, really there's a strong effect of looking at haplotype statistics instead of allele-specific, allele SNP statistics. So founding inbreeding effects are only really detected if we use runs of homozygosity, and we've got these more and longer runs of homozygosity compared to source. And we found it happening more so in a population with a known population crash. And then we've got this really strong candidate of repeated selection at chromosome 15, 5MB, using haplotype statistics, but also using tests specifically for parallel evolution. And we know this region has been selected before in natural populations, naturally colonized populations. All right, so the last major study I wanted to talk about is looking at temporal genomics. So what has happened since 2013? We've sampled these populations again in 2018, and so this is work done by Ali Hudson. I do apologize, different PhD students mean different color legends, but it's the same populations and some of the same data. So we've got our 4 replicate populations now sampled from 2013 and 2018. And we want to know how things have changed over time, sort of demography changes over time. And the nice thing about temporal genomics and sort of method development is that we can now start to test differences in polygenic and single loci dynamics. And I'll go into that in a bit more detail. All right, so first, genome-wide, looking at PCAs again, populations continue to diverge. So we've got our PCA. Each of our 4 populations are kind of going out from the source in the middle. And in triangles, we've got 2018, and in circles, we've got 2013. So some populations are more diverging than others, but they're all kind of going in their own trajectories. Similar results are seen at FST. So if we look at genome-wide FST, it's slightly higher compared to source in 2018 than compared to 2013, but not massively so. So we've got this general drift pattern happening over time. What was interesting is we did that test of inbreeding with runs of homozygosity, and we found that the evidence of inbreeding is quite transient. So what was once really a striking difference in 2013 is no longer there in 2018. So by 2018, all of our populations have lost that large class of runs of homozygosity, and they quite resemble the source population in 2018 compared to 2013. Even in our really crashed population, Taylor has started to look more like the source population. So these effects of inbreeding are quite transient. They're no longer, say, a problem in our populations, despite population changes over time. Okay, so testing for polygenic selection. So here we're using this method called CVTK from Buffalo and Coop. And this method is really based on the prediction that genome-wide allele frequency change should covary, and that's your test of polygenic selection. And we can do it because we've got replicates. We can do it over time intervals and within a population. So we can look at covariance between populations in that first time interval, so that from the source population to 2013, and look at covariance between population 1 and population 2. We can do a similar thing in the second time interval, so 2013 to 2018 in population 1 and 2. And we can also look within a population over time. So is there a covariance genome-wide within one population from source to 2013 and then 2013 to 2018? And these are tests of polygenic selection that we haven't been able to do before. Okay, so these are the results. So in the violin plots there is our simulated distribution of just neutrality. And in the points are our observed data, and this is the first time interval. So in the first time interval, all the observed data is slightly higher than the simulated data, but only in a few comparisons is it within the 90, 95 percentile. So only really significant comparing some of the populations at the first time interval. However, if we're thinking about sort of link selection and polygenic selection, maybe we can drill down into this a bit more. And so what I did is I looked at chromosome level data. So this is, again, the covariance in that first time interval for each of our comparisons. So six comparisons for each chromosome. And what we can see, which is a nice confirmation of our other work, is that chromosome 15 pops out as really significant across many of these comparisons, way outside of the simulated region and above 95%, above the 95 percentile in 5 out of 6. We can also see it happening at some of the other chromosomes, specifically chromosome 1 and chromosome 21, but not as repeatable. So there's some evidence of polygenic selection happening at the chromosome level, I think a bit stronger than when we look genome-wide. If we look at that second time interval, the results are weaker. So again, the observed data is above the mean of the simulated data, but well within that sort of chunk of simulated data. And similarly, if we look at chromosome level over time, there's some evidence of chromosomes really being more more covariant than the simulated data, but it's not as repeatable. So only chromosome 20 shows some repeatability. So 4 out of our 6 comparisons was at above the 95% interval. So perhaps not surprising, if we look within populations, we really don't see evidence of polygenic selection. So there's no covariance within a population over those 2 time intervals. Both genome-wide and then looking at chromosome level, most of them are really within the simulated distribution. Only one of the populations do we find any sort of significance, and it's not really, really that strong. So we can say that polygenic selection maybe is happening in that first time interval, but probably not as strong in the second, and certainly there's no overall covariance happening in our populations. Okay, so what if we look at repeatability of large allele frequency change? So here I'm taking that, again, that overlapping outlier approach, finding allele frequency change that is higher than simulated allele frequency change, and doing that in the first time interval, the second time interval, and then across that whole time, so source to 2018. And then looking to see where there's overlap in our 4 populations. So in the first time interval, we've got 84 windows that are showing repeatable allele frequency change in at least 3 out of our 4 comparisons. That number drops to 21 in the second time interval. And then if we look over the whole time, there's some evidence of shared repeatable selection. So kind of similar to the polygenic change, that repeatability is really just really stronger in that first time interval compared to over time. What I was interested in, what we were interested in is sort of the dynamics of the allele frequency change for these candidate windows. And we wanted to see whether things are changing more in the first time interval, more in the second, or are they changing in opposite directions, and how does that look over time? So these are sort of my prediction slides, a bit confusing. So these are all, in A, we've got all the candidates for our first time interval. So these are our selection candidates. And we've got change in first time interval on the X and change in second time interval on the Y. So if the change in the first time interval didn't change in the second time interval, it should overlap with 0, right? So these blue pops on our predictions. If change happened in the first time interval and in the second time interval in the same direction, we should see these red dots. So these are, oh, sorry. I'm just going to get up the presentation. Sorry about that. Oh. Right, so basically we're looking for overlap of 0 either on the X or Y, depending on what time interval you're looking at. So the blues being things are only changing in one time interval, and the reds, same direction, greens, opposite directions. Okay, hopefully it doesn't crash again. Okay, so this is our actual data, just showing for one replicate because they're quite similar across replicates. If we look in that first time interval, we see the majority of the data overlaps with 0. So the bright colors are more of the data than the dark colors, the density plot. So you can see in the first time interval, there wasn't much at those loci changing again in the second time interval. And that's sort of what you would expect if there was a fixation of alleles or a plateauing of allele frequency change in that first time interval. In the second time interval, There's not as much overlap with zero, so the zero happening on the zero happening on the x-axis. So what that means is that in the second time interval, change tended to be in the opposite direction as change happening in the first time interval. And that might be what you would expect if there was some fluctuating evolution happening, fluctuating genomics happening. And finally, if we look sort of selection happening over time, so that's from source to 2018, they tended to happen mostly in the first time interval with some moderate change happening in the second time interval in the same direction. And sort of that's what you would expect if there was that first big jump and then sort of a smaller change over time. Okay, so to conclude that work, we've got demographic founding events and population crash. Those effects are really only present at the first interval. So runs of homozygosity, number and size resemble source populations now. And selection both at the chromosome and single windows are more repeatable in the first time period than compared to the second. And we see that both at sort of the polygenic and single window effect loci. And that's probably what you would predict based on just sort of natural selection theory, that when populations were first introduced into these environments, they were much further away from the optimum. So they had to take those bigger steps, have that bigger selection response in that first time interval compared to that second. We can maybe start to get at some more complex dynamics, looking at multiple time points, but that evidence is still sort of not as strong, yeah, than that sort of first effect. Okay, so in the last just few minutes, I wanted to talk about some ongoing work that we have going on and how this fits into some other work that we're doing. So I talked about how we have these older translocation experiments, and we do have genomic data for each of these older ones, so from 1981, 78, and 56. So we can start to look at questions about sort of that middle long-term change. And because they're coming from different drainages, we can start to understand a little bit more about starting genetic variation. So just preliminary work right now, looking at El Cedro in blue and Arepo in orange, is that they look quite different in terms of diversity. So the introduction population, so we've got AP intro in the light orange and EC intro in the light blue, have quite different distributions of diversity. So pi on the left and Tajima's D on the right. And what we're seeing is the El Cedro is really quite, has a quite reduced diversity compared to natural populations and compared to the other intro. So what I would like to do is look at effective genetic load and sort of things like genetic purging and things on that sort of middle time point of evolution, and how does that affect repeatability. Other ongoing work we have that I haven't talked about is how does this known selection loci, how does this link to phenotype? So the population genetics is quite powerful for finding evidence of selection, but it doesn't really inform on how that's affecting the phenotype, so sort of that top part of the triangle. So we've done a little bit of QTLs, quantitative trait loci for life history traits. We've also looked at color variation using some iso-Y lines, and we're finding that's quite a complex genetic architecture, probably the Y chromosome interacting with autosomal loci. And we've got some ongoing crosses, just finished up those crosses in the lab and genotyped them using sort of more multi-family and multi-generation cross design. So really trying to get more at this Y chromosome stuff. The other area that we're really interested in that I didn't go into today is balancing selection. So we know that these populations are under quite strong negative frequency dependent selection. And Josie's done some work on looking for repeatable signals of balancing selection across populations. And we find these signals of balancing selections even in populations with really low genetic variability. We've also got some real experimental lines happening now where we've varied balancing selection in the lab. And what generally we're interested in is how does this link up with what we know about directional selection in the system. Right. So putting it all together, what have we what would we learn? So we've got some evidence of single locus convergence, with the caveat that really standing genetic variation is really important, and we've probably got multiple haplotypes working there. But we've got two really great candidates to explore: the chromosome 21 that's happening in the corony drainage, and then the chromosome 15 locus that seems to be both selected really early on in selection and appears to be selected in naturally colonized populations with probably a multivariate, multi-haplotype response. We've got evidence of polygenic convergence happening. So we've got that evidence of convergence happening at the pathway level. And we've also been able to detect some early polygenic response across some chromosomes with our experimental populations. And sort of to think generally about what does this mean for natural selection, just a thought that maybe this mix of polygenic and monogenic trait architecture is really important for repeatable and rapid evolution. So we know that large effect loci are easier to select for, and then they're also a barrier to gene flow. So they're being able to maintain. But having polygenic traits might facilitate rapid evolution as well. If you've got lots of variation to select on to get to the same phenotypic outcome, maybe polygenic selection is important as well. Okay, great. So just with that, I'd like to thank all the past and present members of my lab who've done the work. I think I've mentioned all of them at the appropriate time. And then, of course, collaborators who've done a lot of the sort of more ecology work and collecting work as well. And with that, I take any questions.